Oh, I didn't have the audio on. Sorry about that. All I said was good morning and good to see some faces. Welcome back. We are looking at the question of localism, and we now know that at least we can say that some areas of the brain are required for or essential to certain cognitive tasks. Uh, and so here is a brain mapping that you don't want to take too seriously. We don't have clean borders like this that that everyone's brain follows. But this is the general idea. So we have a visual area back here. This is the occipital lobe, where a lot of vision gets processed, but not only here. We have Broca's area around this area where damage to this area results in a condition like Sarah Scott's. And then over here, we have Wernicke's area. Damage here results in a condition like Byron's. And we watched those videos last time. Uh, we have areas where motion is processed. We have the prefrontal cortex, which involves inhibition, judgment, executive functions. There's still a lot we don't know. And the motor strip is right here, number 3. And then behind it is the sensory strip, which we saw looking at face on, motor and sensory areas. And this is called the cortical homunculus. And there's actually two homunculi. There's the motor homunculus, and that's I don't want anyone to think that the motor homunculus is on one side and the sensory homunculus is on the other side. That could be misleading. So notice that the strip goes all the way across the two hemispheres in both cases, and they just happen to use uh, this hemisphere over here to describe the primary motor areas and to draw this homunculus, and then they use the other one for the sensory. Okay, and so going back to this, so you have a strip where the motor functions are here, and then just behind it you have another strip for sensory. And the homunculus just means little man, and it corresponds to how much area of the cortex is devoted to that body part. And so you can see that the face is huge. No surprise, given that we're social animals, and it really depends what we're expressing. That our ability to survive has a lot to do with that. Other animals that aren't so social, like a snake, right? They're just not going to have much going on with their face. And then the tongue, a lot of area there. Huge hands, so a lot of area devoted to our hands, of course. Um, and then very little devoted to the arm, the leg, the torso. And I mentioned the homunculus statue that you can see different versions of. So I just grabbed one here. The hands are huge. The mouth is huge. We're just, our brains are just devoted to the sensations and the control of the sensations coming from and the control of the hands and the mouth. Uh, Good. And we saw 2 of 3 videos. I started the third video, and it's a video where they're stimulating an area that's approximately back here behind the ear, um, called the fusiform face gyrus, also known as the fusiform face area. And they usually call it just the FFA. And I was looking for a good image for you and hadn't decided on which one I liked the best. Um, but the area is right in the back, kind of on the lower part of the brain. Okay, and so uh, they've been able to stimulate brains for decades, and yet we're it seems like we've uh, it, it seems like we moved quite cautiously into this. And so some of this research goes way back. In fact, there seems to have been more research in the 60s and the 70s directly stimulating brains. Um, but it's been going on ever since. And here's a recent case where they stimulated this patient's fusiform face gyrus. And that's the area that seems largely devoted to faces, but not only faces. It turns out that it gets stimulated when you look at letters as well, like symbols. Uh, and uh, so they're stimulating this area, and normally, naturally, the area only gets stimulated when you look at faces and things like faces, including characters, symbols, letters. Apparently, um, I say apparently just because that's what the research is showing. I'm not uh, saying we just have this impression. Uh, So normally you look at these things and the area gets stimulated, especially when you look at faces. Well, now what they're doing with electrodes is directly stimulating it. And it's going to take states that it would very probably not take in, in real life. Okay. So by directly stimulating your brain, you can uh, create conscious states that you wouldn't normally have. Here we go. Oh, let me set it up for a video.
Look at my face. 1, 2, 3.
You just turned into somebody else.
Tell me
Your
about
face metamorphosed. Your nose got saggy, went to the left. You almost looked like somebody I'd seen before, but somebody different. That was a trip.
Could you see my eyes in place?
I could see your eyes, but you could have been somebody else who had, well, he has similar eyes to Dr. Parvizi, but you were someone else. Your whole face just sort of metamorphosed.
Metamorphosis. Did it get warped?
Yes.
Did it keep its own features?
No, it's
Or
almost
shape?
like the shape of your face, your features drooped. Yeah. Sort of like they drooped to a particular direction, kind of stretched and drooped.
All right.
Okay.
Ready? 1, 2, 3.
Yeah, it metamorphosed again, and you look like someone I've seen before, but maybe a different person in my memory. Almost like your nose kind of shifted to the left a little bit, and your look just changed.
It did.
Yeah.
No.
Tell me more.
Well, I mean, I don't have a photographic memory, but you turned into someone else. You looked like someone else.
Did I keep my gender?
Yeah. Oh, yeah.
How did you know I'm not a female?
Because you're still wearing a suit and tie.
Oh, you could see the suit and tie?
Yeah. Only your face changed. Everything else was the same. Yeah.
Did the skin color remain the same?
Yes. Yeah.
Did the position of my lips and nose and eyes stay the same when they got warped?
They shifted. Let's say they shifted to a side and maybe stretched.
So, what are we learning about this area? Because he says Dr. Parvizi's face shifts, warps, changes, skin tone doesn't change, his outfit, what he's wearing doesn't change. It's just the shape of his face. We seem to have an area dedicated to face recognition, right? And then it that seemed to spill over into symbols, as we will see. And what they're disrupting is not simply the visual field, right? Because you know the the outfit that he's wearing is identified as not it's not the job of the fusiform face gyrus to process, right? The face is interpreted to as something to be processed. Okay, I'll let it go.
but they didn't get larger or smaller.
Okay.
It was more of a perception, how I perceived your face. You know.
Interesting. Tell me more.
That's about all I can say. It just all of a sudden you were you, and then you weren't you. You could have been someone else who was standing there in front of me.
Ready?
Okay.
Tell me if the same thing happens again. 1, 2, 3.
Nothing happened that time.
Nothing happened. Are you sure?
I'm sure.
How about now? 1, 2, 3. Okay.
Yeah.
The same thing happens?
The bottom of her face sort of metamorphosized up, kind of stretched up to give her a different look. It wasn't pretty.
Okay.
There's writing on the balloon.
Did anything change?
Something did change for a glitch.
What did you feel?
It felt like the TV moved and then the balloon moved to a certain extent,
I see.
but it wasn't very clear. It didn't stay there. It just
Got it. Could you still understand if it's the TV?
I mean, it happened so fast. I mean, it just something different happened.
Did the shape of the TV change?
Not necessarily. Almost like it just shifted a little bit.
Okay. Read again.
Hope you're feeling better.
Okay. How about now? 1, 2, 3.
Hope you're feeling better.
Did anything change?
Very slight when I first read the word hope. I know it doesn't describe a whole lot, but when something doesn't seem quite right, that's sort of how it felt when I read the word hope.
Look straight ahead in my face and tell me if you still feel that metamorphosis you were talking about. Okay?
Okay.
Now?
No.
You're not feeling it?
No.
How about now?
Yes. Your face started to skew to the right and protrude.
Okay. Perfect. Thank you. All right.
But so far, the most significant change has been on your face.
So that's important because remember, they do the sham trials uh, to make sure that the patient isn't confabulating or playing along with what the patient thinks the researcher wants, which happens. And it happens enough that they have to do these, they have to blind the experiment. So the patient doesn't know when the stimulation is actually occurring. And so, of course, if during a sham trial, the patient goes, "Oh, yeah, I think I did notice something going on with your face," even though they're not trying to deceive, they might do that. Uh, well, then you have to worry that there's, it's the power of suggestion. But if instead, as we see here, during the sham trial, the patient says, I didn't experience anything, and then during actual stimulation, the patient does experience something, that's a very good reason to think that it's a real causal link. And so we also know that there are people who have damage to this area, and they become not blind to the visual world, but they become face blind, prosopagnosia. They they look at there's a book by Oliver Sacks that you might have heard of. It's called The Man Who Mistook His Wife for a Hat. And Oliver Sacks was a neurologist, and he wrote up these little anecdotes. These are like vignettes of his experience um, with patients. And in one case, this couple comes in, a man and a wife, and the wife says he's otherwise normal, but he's having trouble seeing people and recognizing people. And so they had their discussion, and at the end, when they both stood up, the man turned, he'd put his hat on a hat rack, and he turned to his wife, and his wife was staring at him, and he puts his hand on her head, and as if he thinks her head is his own hat. He failed to recognize that he was looking at her face. They can see the face. They can describe the color. They can describe the two round dots, and they can infer those are probably eyes, right? But they can't process the face and understand the face, and certainly they can't recognize people. And they sometimes take things, according to this story, the man who mistook his wife for a hat, he would sometimes say something to, apparently his wife saw him talking to or saying something to a fire hydrant as if it was a child, right? So he might have had a more degraded brain. He might have had other problems in addition to problems with the face area. Any questions or uh, comments? You have the book. Yes, Emily, it's a great book. He Critics say he takes some literary liberties because he writes as if he has a perfect memory of the dialogue, and the dialogue is very literary. So it seems like fiction, but it's all based in truth. Uh, and I see Alejandro had written, for some reason, this was making me think of lobotomies, removes certain abilities. Um, Illness per se. I liked how much people might improve with a correct current of electricity flowing through our nervous system. They do sometimes get improvements. I don't know why this patient has had brain surgery. It could be for epilepsy. They often put in electrodes to try to control or treat epilepsy, and it can work, doesn't always. Uh, and it could have been maybe some brain trauma, um, car accident or something. I really just don't know. Yes, Darla, that's wonderful. Have you seen the video of someone playing a violin during brain surgery? Yes, I have. And does anyone, I mentioned why this would be the case. Why would the patient be awake during brain surgery? Why would you do that? Anyone know? Yes, Yasmin.
I think it's because it's kind of one of the only ways that they want to. It's so hard to operate in the brain, right? And understanding like how it's impacting different parts of the brain. I think that was part of it.
Yes.
Right. And yeah, I was going to say something about pain, but I'm not sure if that has actually any correlation to.
Well, yes, Yasmin, you're right. Uh, I say don't take these maps too seriously, like the one you're looking at right now, because we can't just go from brain to brain and say, okay, right here is the borderline around Broca's area. So they really do poke around. They stimulate, they anesthetize, and they try to figure out where do these areas start and stop. And if I excise something, if I cut something, will the patient lose an important cognitive capacity? So this woman played violin. I believe she played it for a living. She was in an orchestra and all that. So it is crucial that she not lose her ability to play violin. And that's not just the ability to move your arms and fingers in a certain way. It's also cognitive task of playing music, is very demanding. And if when they anesthetize a certain area, she's playing but very poorly, then they can reasonably infer that her her ability requires that area. And if they anesthetize another area and suddenly her right arm is uh, effectively paralyzed, right, then they know that's an area that they should try their best not to excise, not to cut out. Yeah. And uh, iPhone, I don't know who you are, but I see your hand up. Go ahead.
I don't know if that's me. It might be me,
Oh, that was
actually.
me. I'm sorry.
Oh,
I'm on my
sorry.
phone. This is Jerome. My bad.
Okay,
But
Jerome, hi.
yeah, I wanted to say, I was asking, is there any truth to the idea that like a certain part of the brain could be like bigger and therefore like that person is like stronger in that area? Like, like if say like your frontal lobe is like, larger than somebody else's, that means you're, I don't know, better at addressing certain
Well,
types of situations, or if it's smaller, like, is there any
yeah.
truth to that?
Well, okay, so you got to be really careful. I guess very roughly, there's some truth to it. So, but very roughly. So it's not like you can look at a patient's brain and see, oh, this patient has a huge frontal lobe. They must be a genius. I'm not aware of any research that shows that. But if I did mention the cab drivers of London, and certain areas of their brain had uh, more connections, and so that on a neural level, and we're about to learn about neurons, brain cells, right? Uh, they had more connections, and you can do this with athletes. Their cerebellum, it has more development, so you can, and even the nerves running down the spine and so on, which isn't that surprising. But like a gymnast, someone who practices gymnastics day after day after day, as they develop, right, their brains will show that difference. So there's at least that, and of course, if an area is damaged or underdeveloped, then they're really straightforward cognitive deficits. So some humans come into the world with underdeveloped brains, and sometimes extremely underdeveloped. But so much so that some people think they don't even have any conscious experience at all. And so, but in other cases, I mean, there are also patients who, for all we would think, they seem totally normal. Um, they go to school and they get jobs and have families and whatever, and then for some reason or other they have to do a scan and they discover there's like a big chunk of their brain missing. And that's interesting. We have these areas that do seem dedicated to cognitive capacities, but it's also the case that sometimes damage to those areas or underdevelopment of those areas results in neighboring areas uh, taking up the slack, and learning the cognitive capacity after all. Grace writes, this conversation reminds me of Gall's phrenology and how his beliefs kind of made me think of eugenics. Right. I teach another class called Ethical Issues in Science and Technology, and we discuss eugenics. Uh, and there was uh, a lot of interest about 100 years ago in using the biology of the brain and what we're learning about biology in general, um, well, people had different ideas, but some people thought we should improve the human race. And maybe by studying different brains and coming up with some kind of science of what makes people smarter and more athletic and all that, more artistic, perhaps even. Maybe we can try to breed a better population. Uh, and it has a pretty dark, twisted past. There was a lot of what they now call scientific racism built into it, and it really started in America and the UK, and then was taken up by the Nazis. But they were inspired largely by the Americans in their in this regard. Um, yeah. Uh, So and so phrenology was part of that, just to tie that up. So Grace, you're right. Other thoughts or questions?
I knew someone that back in 2002 had brain cancer and had an operation at a cranium. The surgeons were trying to see what they could do about the cancer cells. And so he was this person was not awake when the surgery was happening. But afterwards, this is an example of where they probably inadvertently touched something in the brain that changed him when he recovered from surgery. And it was in the prefrontal area, the left, I think it was, and his sort of inhibitions and the things he would say. And it was kind of wild. Um,
He became way less inhibited.
yeah, and it was sort of like, what is going on? We didn't know. And the surgeon was like, well, it's possible that this happened. So yeah, it's a real thing.
Yasmin, there's a famous or notorious case of a man. There are a few cases like this, but the one I have in mind, I don't remember. I don't think we know the patient's name, and I don't remember how to refer to it. But uh, he had lived up until a certain point a totally normal life, didn't commit any crimes or anything, and he just suddenly started acting very inappropriately around women, including his own family members, and it just became very creepy and weird. And I don't know how far it got, but they finally looked at his brain, and he had a tumor right here, frontal left lobe, which takes away your inhibitions. In his case, they excised it, they took it out, and all that behavior went away. A few years later, it started to resurface, and sure enough, the tumor had returned, and they had to do uh, brain surgery on him again. The There was a shooter, I think it's the Texas Tower shooter, who shot a bunch of students long ago from a tower. I think that's the one who wrote, when I'm dead, look at my brain because I think there's something wrong with my brain. And sure enough, I don't remember if it was a tumor or something else, but um, he had damage to a part of his brain. So yeah, you know, you may not even be conscious of the degree to which you inhibit things. I mean, we're all conscious of it to a certain point. Um, but we will see later that I think we need to um, take the fact that you can temporarily even lose parts of your brain and gain them back due to something like anesthesia, or a tumor that gets excised, that gets taken out. That I think says some really interesting things about what we are as conscious thinking beings. So we're going to get there. We're going to learn more of what we're doing today, but we're also going to learn about split brains, patients who have had the bridge between their hemispheres uh, cut. And you can't see it here, but about here you have this bridge called the corpus callosum. You also can't see it here. It's covered up. It's deeper. Here it is. This thing. Uh, at least I think it gets thinner where the hemispheres meet. Anyways, when that gets cut, uh, because it's been cut in patients, usually to treat epilepsy, we've learned a lot of things about how the brain operates, and so we'll learn about that, and then I'll share some work that I've published uh, about how we should think about the brain and its parts. Okay. Oh, and then I saw in the chat, Zaw wrote, I think some people who were born blind or became blind at a very young age develop their senses more in other parts of the brain. Is that called neuroplasticity? Yes. Uh, the so there are different types of plasticity, but neuroplasticity is when the neurons take over jobs that they wouldn't normally have. So if you have a small stroke in an area, and so a cluster of neurons die, we know that surrounding neurons will grow at least new dendritic spines, but usually new dendrites. And what are dendrites? Well, just think of them for now as like the roots that receive signals. Okay. And the brain develops. The brain makes physical changes every day. We know this. Uh, some people are sometimes skeptical of this. So there still is this view, and there used to be this view that the brain only changes slowly over time in a physical way, but it's the process of activity, the activation among the neurons that changes. But guess what? There are people who have fallen into ice cold water and been underwater for sometimes, because it's ice cold, you don't die. Like, so sometimes it could be 20 minutes, 40 minutes, even over an hour before they pull the person out. Now, a lot of people die when that happens, but some people don't. They come back. They come back. And as far as we know, when they're frozen under at the bottom of that lake, while the first responders are trying to show up and go down and dive down and rescue them, uh, as far as we know, there's no electrical brain activity. Okay, so their brain is still physically formed as it is, but it's effectively frozen, and it's not active. The patient is revived, rarely, but sometimes. And guess what? They remember what was happening right before they fell in. Most of them, many of them. All you need is some of them. They say, "Yeah, I remember. I was pulling on the rope and leaning over the boat, and I fell in the water." Right, and that strongly suggests that their memory is physically encoded in their brain within less than a minute. Right. So right now, you're taking this class, we're talking, we're having discussions. You saw a video, now you're looking at an image. Like, that's physically changing your brain. I used to do that. Well, I can do it now. Uh, it, and it's one of those things that can be kind of trivial and maybe obvious, but also uh, kind of magical, right? I can change your brain right now with a number 137. I just physically changed your brain. How do we know that? Well, because many of you in a minute from now, if I say, what number did I utter? You'll say 137, right? And some of you will remember tomorrow, and in some period you'll forget. But we have every reason to believe that's not just changing the activity that the physical brain uh, propagates, enables. See, we seem to have good reason to say that that's physically, dendritic spines are forming, perhaps even dendrites. But I would pull away from that. We at least know that within seconds, sometimes dendritic spines are developing in the brain. Uh, and that's got to be, according to our best theories, physically encoded. Uh, Not everyone believes that. Some people think it requires some kind of immaterial uh, mind. Okay, Yasmin, go ahead.
So if memories are physically encoded, all memories, whether or not we
Hmm.
can conjure those back up, I was thinking about a story I've heard of certain people that um, literally remember every single, can recall every single thing that
Yeah.
occurs within their day. And it kind of, for them is a good and bad thing, obviously, because to have to remember everything to the degree that they are able to, the detail. So how is that? Like, what is that? Don't know.
I I don't know. I know that there are these people. They get studied at UCI, by the way, who you can ask them to go back many years and pick a date on the calendar and just ask them, what did you have for breakfast? What did you watch on TV? And they can walk you right through it. Now, do they remember like what position they were in on the couch when they were watching TV? Do they remember every detail that you have access to when you're living the event? Like right now, I know what my posture is and so on. I don't know about that, but it's still amazing how much they remember. Uh, and I don't think we have a good understanding of the difference and what it is about their brains um, that allows them to remember such things. I will say this, that some really amazing, fantastic memory feats are accessible to any of us. I mean, if you've made it into college classes, even if you have some cognitive disabilities or impairments, you can probably, if you care to do it, you could memorize a randomly shuffled card deck. You could learn to do that. And I can't remember when we do associationism, which we did. Sometimes I mentioned that you can use that to memorize things. And so, just out of curiosity and for the fun of it, um, I I might have already mentioned this. I started memorizing the digits of pi, and I got to 20, and then I got to 40, and um, I maxed out at 120, but now I think I could really only do the first 100. Um, it's not as challenging as you might think, and it doesn't involve rigorous concentration. Uh, it can, as long as you come up with interesting associations between the numbers, it's like learning a story or learning a song. And so I recommend it to anyone that who wants to just uh, stretch their boundaries, see what their minds can do. It doesn't have to be as challenging as it might think, but it's certainly impressive. And I should say we all do this anyways, right? Um, you can read English, which means you can look at long strings of symbols and recode them as a meaningful sentence, and then later you can unpack that and song lyrics, passages perhaps without effort. Sometimes you remember movie lines and so on, and that can break down into many, many symbols in a certain order. Uh, and a lot of that was done for you when you were learning to read, but we already access this power. It's just since we all do it and we don't really think about what we're doing, it may seem kind of mundane. Uh, Jerome, and anyone can ask me more about that in office hours. You're welcome to do that. Jerome, go ahead, please.
Yeah, I was just thinking about the fact when you said how certain memories can be physically encoded into
Yeah.
our brain, and I think that, I don't know, the idea that like when you're just kind of studying, how you can just be sitting down at your computer, not doing anything physical, but if you're just using your brain a lot, if you're thinking a lot, if you're just reading or writing, how you can feel exhausted afterwards. And
Yes.
think about, even though you may not think you're doing anything physical, but if your brain is actually, if there is something physical going on, then that would explain it. That would kind of help explain that as well.
That's right, Jerome. And actually, we now know that we don't sleep so that our bodies can recuperate so much as we sleep so that we can code memories. It's the brain that needs sleep, not the body. The body can walk and walk and walk for days. I mean, humans have had to do it, right? And sleep isn't what the body needs to recover from a lot of stress, exertion, I should have said. Of course, there's more going on than just the brain recuperating, but a huge amount of it has to do with, according to our best theories, coding short-term memories to long-term memories. Uh, and some people nowadays quibble with the short-term and long-term distinction. There's some research that goes against that distinction, but a lot of people still use the term, and so I'm just pointing that out in case you've heard that. Uh, Good. So, and yes, if you don't sleep, um, if you've gone for a long period of time sleep deprived, uh, you can still be physically able, but you will have mood swings. Eventually you'll have perceptual and judgment problems, and all your cognitive faculties just fall apart. You start to hallucinate. It gets pretty bad. Okay. So, it wasn't so long ago. It was like a little over 100 years ago that we figured out what the brain was. We just didn't know, and we didn't have the tools. And so there was, is it an interconnected network like the circulatory system? Yeah, or do we have separate cells, right? So do we have nerve nets or neurons? Bobby wrote, can you come back from that? Oh, from sleep deprivation? Yes, fortunately. Yeah, if you go for about, so um, there were horrific experiments performed uh, by the Nazis. And there were also sometimes there was a radio DJ in like the 70s who just for entertainment on a dare was supposed to stay up for days. And I think it was day 3 or 4, he just starts falling apart and just blabbing incoherently. And but yes, if you get your sleep, you can come back from that. Uh, I think people can also sense a smell again when they remember it in their mind, as if their memory brings the scent back. Strangely, the olfactory system and the memory system in the brain are very interleaved or interconnected, and sense can be very powerful um, uh, memory triggers. So much so that this is the opposite of what you just said, because you're saying that thinking about something can bring the scent back, and I think that's also true. But sense can bring odors, can bring memories back. So much so that some cognitive scientists have said that when you study, if you use a scent while you're studying, and then when you go to take the exam, you use that scent again, whether it's a perfume or something else, maybe it's sage or something. That they say that that can help. I don't know how good the research is, and I tend to be a little skeptical. Um, uh, Meaning, not that I would bet against it, but I would be I would probably bet in favor of it. But I haven't really dug into the literature. And I'd be very cautious because once research like that suggests something, uh, there's kind of the science writers who aren't really doing science research, and they take it and they go with it, and it becomes a meme and so on. So I would just be a little careful. Uh, there's also some research that suggests that the scent of cleaning products makes people more attentive. And so anyone's welcome to look into that. If you find a link that you think I'd be interested in, please share it. I love stuff like that. There was a replication crisis. There has been in psychology and cognitive science. The replication crisis is there were all these exciting pieces of research, and then as the years went on, everyone treated it like it was knowledge, and then someone repeated the experiment and did not get the same results. They could not be replicated, and so we lost a lot of these fun, uh, research results in the replication crisis when people repeated the experiments, and in some cases they could replicate the results, but in other cases they couldn't. Yes, smells are more tied to emotional memories, like the smell of your childhood home or mother's perfume. Yeah, absolutely. And from an evolutionary perspective, you can look at. I mean, the olfactory system is buried deep within the brain, along with where memories are encoded. So there are a lot of reasons to think that they're closely tied together. Okay, so we were trying to figure out what the brain was composed of. And the neuron doctrine, as it's used here anatomically, is just it's composed of little brain cells. And then the nerve net theory says it's like the circulatory system. So there are a bunch of just a network of tubes through which something must flow. And later they started to think, well, maybe it's electricity, but prior to that, was there some kind of like a gas or something? And we just didn't have the tools uh, to settle this. So Golgi is known for this silver nitrate staining method, and this allowed us to see neurons in more detail under a microscope. And so here you have neurons. This is the soma or the cell body, the clump, and then you have an axon going up, and you have dendrites coming out like roots. And he just misinterpreted what he was looking at. He thought the dendrites supplied nutrients, and he also could see enough to see that they were interconnected, but he couldn't see enough to see that there's often a gap between one neuron's axon and another neuron's dendrite. So where they connect, there's a little gap called the synaptic gap, and he couldn't see that, and so he thought it was an interconnected network, and this supported his kind of the holism because so think of the blood circulating through the body. If the blood gets infected, I don't know, um, you prick yourself and something gets into your blood, um, it within a moment it's throughout your system, right? It spreads, and so some people thought of the brain like that. Whereas if it's composed of discrete parts, then maybe you can say, well, this part's got this job and this other part's got a completely different job. And so the holism, localism debate is being informed by these advances, sometimes misleading, in brain anatomy. So it wasn't until Santiago Ramon y Cajal that we understood that the brain is composed of neurons. He discovered the neuron. And by the way, anastomosis, I should have mentioned what anastomosis is. Um, anastomosis is when, in biology, something branches out and then comes back together. So if tree roots branch out and then some tree roots meet and join together again, that would count as anastomosis. And there are some sea creatures that exhibit this. So they have branching developments and the branches come back together and you end up with something that looks like a net. Okay. Uh, and so the neuron, as Ramon y Cajal sees it, um, is the anatomical, physiological, metabolic, and genetic unit of the nervous system. It's all a bunch of neurons. Of course, there are other cells that are structural, but we in cognitive neuroscience are focused on the neurons. And they all have a cell body, they have an axon, which sends signals, and dendrites, which receive signals. Checking the time. Okay. Good. Okay. So meanwhile, you have Carl Lashley, and he would take rats and put them in mazes of different levels of difficulty. And there would always be a treat. So the simplest maze would be like a T shape, and the treat would always be on the left. And so the rats would very quickly, the moment you put them in at the bottom of the T, they'd run down the hall, make a left, and go get the treat. Okay, so that means that the rat remembers where the treat is. And then he'd have a more interesting maze where they had to make a few turns. And then a very complex maze where they'd have to learn to make a lot of turns. And they would all learn this. And then the question is, remember, we were just talking about memories being physically encoded. For Lashley, the question is, where's the memory? Is it here? Is it here? Is it like where is the memory? So he performs ablation experiments on these poor rats. I don't mind saying that. He basically cuts away parts of their brain. And the effort was to try to locate where the memory was. So he cuts away, this part of the brain, and the rat still knows to turn left. And then he cuts in another rat, he cuts away this area, the rat still knows to turn left. He actually could find no area that seemed to be essential for remembering where the treat was, for remembering the layout of the maze. In fact, I just finally took this from the book because I got sick of drawing it all the time. You can map this out in 3 dimensions. So from the left to right, from 1 to 10 all the way to 51, that's the percent of the cortex that is removed. Okay. Percent of the cortex that is removed. So that's what ablation is, by the way. To ablate is to cut away the surface. And then the maze difficulty goes back. So maze 1 is simple. Maze 2 is a little more complicated. Maze 3, much more complicated. Okay. And then vertically, we have error. Okay. So you can look at the front here. Think of this as a tabletop. And then the higher you go up the table, that's more error. You watch the rat go through the maze, and if it ever takes the wrong turn, that counts as an error. And look at this. For maze 1, even when the rat had 51% of its cortex removed, the error was just slightly higher than it was for 1 to 10%. So you can be missing half your cortex and still remember where the treat is in the maze for maze 1. And then for maze 2, well, there is a difference. You can see it, but it's not much, right? You have to go to maze 3 and cut away a lot of the brain for the error difference to become obvious. Great. And so, of course, the less brain matter you have functioning, the less you can do. So at some point, they do start to exhibit errors. But he could never find the location where the map of the maze was stored. It seems to be distributed throughout the brain in a holistic way. And so we have these terms, equipotentiality and mass action. So equipotentiality, it means they have the same potential. One region can carry out the function of the whole. Even so, when the whole was carrying out the region, and you take away some of that, the remaining region can still do it. Uh, uh, Often in a way where you can't even tell the difference. But for a more complex maze, you will eventually be able to tell the difference. uh, Mass action. The efficiency of an entire complex function may be reduced in proportion to the extent of a brain injury to a region with equipotentiality. The point is that no region of the cortex seems necessary for knowing how to or remembering how to navigate the maze. You know what? I want to update that slide. Because I don't want people thinking that it's about what I had there was um, no single region of the cortex seems necessary for navigating the maze, and that might make you think it has to do with just seeing them just wandering around the maze. But no, we're talking about rats that have exhibited a memory of where the treat is. So that's in favor. So that makes us inclined to be holists about memory. That's not to say that you don't. I mean, there's certain like the hippocampus and certain subcortical parts of the brain are essential to for coding memories. If you have damage there, you will live life in very short moments, constantly forgetting things that just happened. Uh, the old Christopher Nolan movie Memento is about a guy with that impairment. He can't make new memories. Uh, and so yes, there are certain regions where you can localize those capacities. But when it comes to remembering mazes and locations and people, it seems like that's largely distributed over the brain. So it seems like you can be a holist about that. Uh, Sometimes patients lose, as we will see. We're going to discuss patients who have lost an entire brain hemisphere. And sometimes they come out of surgery and the family members want to know, do you remember me? Do you know who I am? And sometimes they just, yeah, mom, dad, I know you all. And um, they have other deficits, but remembering people and all kinds of things is it left unaffected. Donald Hebb is known for Hebb's law. When one brain cell excites another enough, there's a change in A's capacity to excite B in the future. And everyone now says cells that fire together wire together. So if this cell A keeps stimulating this cell B, right, the more often that happens, the more likely it will happen in the future. The more likely its ability to trigger the other cell to be stimulated um, in the future. And this enables you can have these cell assemblies, as he called them, and phase sequences where a cell is stimulated and that triggers like a domino effect, and other cells are stimulated, and then some of those circle back and stimulate the cell that was originally stimulated. And so you can get these kind of self-perpetuating cycles, these self-exciting circuits. And maybe this allows us to kind of merge localism as whole and holism as Harnish sees it, where these particular cells, they're in connection with others. So you can localize their functions. Say this cell is really receptive to, say, edges in the visual field. We have what are called edge detectors, as we will learn. But then they play a role in this more holistic phase sequence. So let me check the time. Good. Learn some real just basics about the neuron. So the neuron has a cell body, also known as a soma. That's Latin for body. And then it has an axon that sends signals. And it sends what they call an all or none signal. It's either firing at a certain amplitude or not. And so that implies that there's a threshold. And uh, you never get um, half a spike or, well, let me see if I have this somewhere in the slides. I don't remember. Yeah, I don't. So I'm going to draw what you never get. So if you look at a spike train, as they call it, which is recording what a particular neuron is doing, you'll see something like spike, and then it dips down, and then spike. And that's the refractory period. It'll never, I don't think you can see my cursor until I do this. Okay. Uh, you can make these you can have a higher frequency, right? You can crowd these spikes together. So that's continuous, but the amplitude is always within a tiny little error margin. It's always the same. And so you will never get, trying to get my pen back. Okay. What you'll never get is this, like half a spike. And you'll never get twice a spike. Okay. So I'm crossing that out. And that's the all or none. So it's um, you know, you could imagine this being encoded with ones and zeros, right? So it's like 1, 0, 0, 1, 0, 0, 1, right? All or none. The dendrites receive signals in a graded fashion. And so the dendrites, I'm going to erase all this, uh, will um, sum the graded signals that come in, and basically the cell body has a threshold, and if you have enough um, electrical activity, let's just say, if it meets or exceeds that threshold, then a signal goes out along the axon. And again, that signals every spike is all or none, but the frequency can be high or low. So you can imagine one neuron that's going tick, 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 and another one's going tick, 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 right? And that's a difference of frequency. And these things look like this. I see something in the chat. Right, we're learning now about action potential. Thanks, Layla. And are these all different types of neurons? So we do have different types of neurons. And yes, as far as I know, so any neuron that whatever its spike amplitude is, as far as I know, it stays relatively fixed. Now, might there be two different neurons with two different amplitudes? I think so, but that's a good question. I don't know for sure. But the point is, it's all or none for any particular neuron, at least. And yes, there are different types of neurons. So a typical neuron looks like this. It has one axon, and if you want a mnemonic to know the difference between the what the axon does and what the dendrites do, think the axon. I'll put this in the chat. Is the one that sends the signal. So the axon acts on the dendrite. Okay. And that is it sends a signal. Now, the signal can go to another neuron, and we call those interneurons, but it can also go to a muscle and it cause the flexing of a muscle or an organ, right? And the dendrites are receptive usually to the signals from other um, neurons. So here are the dendrites and the dendritic branches, which have little spines, which research at UCI long ago showed that you 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 develop dendritic spines sometimes within a minute. So the idea was maybe that's how memories are. Maybe that's part of the story for encoding memories. Uh, Here's the body, also known as the um, uh, the soma. And there's some things you don't need to know about, like the endoplasmic reticulum or the mitochondrion. Basically, just the axon, the synaptic gap or synaptic terminals. We're going to zoom in on that. The dendrites, and that's it. Okay, so here's another visualization. We have the signal going along the axon, and then it gets here. And then typically you do have a tiny little gap. And once the signal reaches this synaptic terminal, there's this little gap, and you have neurotransmitters here. And there's like an explosion, like a volley of transmitters go across that gap, across the synapse, which is the gap, and get received here by receptors. And then a class where you learn basic neuroscience, you learn about different types of neurotransmitters and all kinds of other things that we're not going to get into. Oh, and once they go flying across that synaptic gap, they're typically taken back up. It's called reuptake. And so you get things balance out and they go back. Um, the uh, the nerve impulse, also known as the action potential, which the name action potential has always bothered me because we're talking about an actual event, a signal going down an axon, right? But they call it the action potential. It's also known as the nerve impulse, and it's an electrochemical signal. It's not just electrical. It's very slow from an engineering standpoint. Our electric circuits are way faster, and then we have fiber optics, right? And transmission at the speed of light through the electromagnetic spectrum. Uh, and and so, as some evolutionary biologists will point out, it's for organisms to live in real time. Brains have the incentive uh, or are at an advantage if they do this, if they distribute problems and process them in mass parallel fashion, as opposed to in a serial way, because we process so slowly. So we don't want to solve problems in a serial manner. We want to solve as much of the problem in parallel as possible. Uh, the nerve impulse or the action potential can excite a neuron, stimulate a muscle, or activate a gland. I mentioned I drew the refractory period. That's I'll draw it again because I want to make the point. Uh, so here's one. Let's make it a very low frequency. And then during this refractory period right here, it doesn't matter if a lot of inputs coming into the dendrites, it will not stimulate. So the refractory period is when it's um, not going to respond to input. And so the highest frequency is right after that refractory period. I didn't mean you could dip it down so low, right? The refractory period should be the same in every case. So you can crowd it up to the point where the spike is close to the refractory period, and then you can't make it any more crowded. So you can't have a higher rate frequency. I'm going to erase that. What else? Often, most of us are in the bad habit of talking about one well, and one brain cell neuron stimulating another and activating it, when in fact they also inhibit each other. So the signal doesn't always cause the other one to fire. It can do just the opposite. You have inhibition. And almost we just are always in the bad habit of talking as if that doesn't happen, even though we all know it does. And then finally, summation is where the signals from sensory surfaces and other axons, right? So you have neurons in your retina, and they're responding to photoreceptors that are responding to light, right? But then in your brain, you have neurons that are responding to other neurons. And when they get that input, that gets summed or added up, and that will may or may not reach a threshold and determine whether the receiving neuron will fire. Okay. Now, the book, which you're no longer required to read starting this semester, has an error on page 75, and it treats potassium ions as if they have negative charge, which they don't. But this image is still accurate. So you have a gradient. You do just have a difference of charge. And so when the neuron is in what we can call a resting state, you have positive sodium ions on the outside, and you have a more positive charge on the outside, and negative, relatively negative charge on the inside. And then you have these gates. And when the activity in the cell body reaches a threshold, these gates right here open. Okay. And the positive sodium ions, which are outside, they rush in. And the quote unquote negative potassium ions move out. So you get this exchange. And we'll have some videos that are optional that go into much more detail, created by our TA, Banaz. And one thing she does at the very beginning of the video, she has a wonderful mnemonic for remembering this. Just think of a banana in the ocean. The banana is potassium, and it's in an ocean of sodium, right? Salt water. And that's the default state. You have the potassium inside, the ocean of sodium on the outside. And I like to think of this as being on Muni or BART, I guess, whatever. And there are those outside, they can't wait to get in, and those inside can't wait to get out. The moment the doors open, you get that exchange. Now, the difference would be, what if the next set of doors would only open if the previous set had that exchange? So the doors don't all open at once. These gates open and let that exchange occur only if it's happened previously. So as you can see here, it's halfway through this process. And this is like a domino effect, because notice that nothing's being sent down a tunnel. You don't, it's not like blood coursing through the veins. It's these gates open and you have this exchange, and that causes the next set of gates to open, and then you have an exchange, which causes the next set to, right? So it is like a domino effect, where when dominoes fall, you don't have a single domino going flying down the road, right? Each domino just does this. And here, each gate just opens and you get an exchange of sodium and potassium, and that's it. And so the signal is, it's potentially, there's no point at which it would die out or fade, right? So that's something that people don't usually talk about. Okay, any questions? If there aren't questions, then what I will do is set up where we're headed, which is we're going to learn about split brains. Don't look at that. Look at that. Okay. We'll get there. And this is, for a lot of students, one of an exciting part of the course. Unfortunately, I don't think we have time to start a video, but we will um, do that next time. I'll make something available over of course, over tonight or tomorrow morning. Um, Here's what I'll say right now. So you have two hemispheres in your brain, and they're connected by this thing called the corpus callosum right here. And there are other little bridges, they call them commissures. But the main one is the corpus callosum. And there is also what's called the optic chiasm or chiasmus um, below the two hemispheres. So that's an indirect bridge. We're looking down at a brain, and there's the left and right halves of the visual field. Of course, we have a lot more um, peripheral vision than this suggests, right? Way out here, okay? It's not like we have tunnel vision. Um, and notice what happens. The it turns out nobody knows why this is the case. There's some theories, but it turns out that the left hemisphere is oh fun, Araceli. I don't know if it's one that I will be sharing or not, but I'll click on it later. Um, Actually, what I'm going to do is copy it. Okay, thank you, Araceli. Uh, so the left hemisphere is largely in control of the right half of the body, and vice versa. The right is largely in control of the left. Remember, Sarah Scott got pins and needles in her right arm and leg, and that suggests damage in the left hemisphere, which is statistically for most of us where the ability to produce language is located. Uh, okay, and the same goes for visual. Our visual system is lateralized. So notice that the dark shade for the right half of the visual field, it goes into each eye. It doesn't go into one eye. So this is a misconception that understandably a lot of people have. It goes into each eye, and then it goes directly back to the left hemisphere. And then what's going on on the left goes through both eyes and then directly back to the right hemisphere. And then very quickly, unless you have a severed corpus callosum, 3 milliseconds, that information crosses the corpus callosum and ends up on the other hemisphere as well. So we have this weird crisscross where what you're seeing in your visual field, everything on the right goes immediately back to the left hemisphere. Everything on the left goes immediately back to the right. But within just a few milliseconds, that all gets transferred. And you have no idea any of this is going on, right? Here I say LGN, that's what they call the lateral geniculate nucleus. We'll get to that later. Now, the question that we're going to look at is what happens when you cut that connection? And so the information on the right half of your visual field goes back to your left hemisphere and never gets over to the right hemisphere. So the right hemisphere never knows what the left hemisphere sees. And the same goes for the left hemisphere. It never knows what the right hemisphere sees. And what does the left hemisphere do for most patients? It produces speech. So it can tell us what it sees and what it doesn't see. So that's what we're headed. That's where we're headed. Okay. Really strange, fascinating stuff. That's all for today. It's 10:45. I will announce what I'll have you do for next class. I guess one short video is probably what it's going to be, and some links, I think. And we're getting into split brains. All right, that's all for today. Thanks, everyone.
Thank you.
Thank you.
Yeah,
Thank you.
I want to see what Araceli's video is. Ending the recording.